Yes, I did check everything like this. Okay, perfect. Thank you so much for joining me today. We can maybe still get that door closed so that there isn't too much background. Okay.
Some air.
Very well. Okay, my name is Robin Moore. I also do metabolomics, and I thought that was absolutely great to have Hannes, who I've been a big fan of, present before me, because I think that what I am involved with and what Hannes is involved with are both two sides of the same coin, and they're really, they complement each other very well. So like, you know, what Hannes is doing is basically saying like, okay, right, with these 123 metabolites in a panel, we can predict a lot of diseases. But some diseases are really complicated to understand biologically what they even are. And so in order to be able to create these panels and be able to say, okay, yeah, with this panel, we can reliably predict the disease, we sort of have to understand the disease itself in like some kind of way. For example, what are we even looking for? What is even being disrupted and what's being like what's, you know, actually happening in the body, and if you don't have any like exploratory mechanism for finding new things happening in the body, for example, like this glycoprotein A, which Hannes was mentioning, then you potentially are at the risk of not actually seeing disease-specific markers. So there's actually like disease-specific research that's required, and that's what I'm going to be talking about. So I'll be talking about breast cancer in dogs, which is referred to as mammary cancer, and I have been studying that on a metabolomic level. So we're luckily and thankfully already quite aware of what metabolomics is, so I'll keep the introduction and background and text short. I've already have presented somewhat extensively on this topic, and indeed I have just been finishing up this manuscript, and I'm just trying to get it submitted at the moment. So this is the final data. Finally, after many years, I am able to move on to new projects. And so this is sort of like a summary of what I learned from this project. And hopefully, if I have time, I can look at the future research directions where I'm going after this study. So in any case, if we like just start with the basics, dogs, like humans, suffer from mammary cancers.
Can you take the top thing off?
Okay. Maybe. Hi.
Thanks.
Okay, so in any case, there are certain risk factors that have to do with mammary cancer, and that has been established in epidemiological studies. For example, the Samoyed dog is... yeah, so for example, the Samoyed dog is cute, but also has a genetic predisposition for this disease. So there's like certain things that have to be taken into account, but it gets tricky because mixed breed dogs are, even though they're less susceptible to mammary cancer, when they do get mammary cancer, the chances of it being deadly are way higher. So that's the same thing with, for example, small dogs versus big dogs. Big dogs are at a risk for developing more deadly disease, while the actual incidence rate is much higher in small dogs, and that has to do with being old. You and simply through age, if you're an old dog, chances are you're a small dog, and if you're an old small dog, chances are you'll get mammary cancer at some point. So there's these things that are like background factors, but then diet really hasn't been considered in any meaningful way when talking about this disease. So this is something that we want to study as well. And then obviously we want to just have a more holistic environmental approach of understanding what it is in our environment that seems to be causing the incidence of mammary cancer to not be decreasing currently, and that it applies for humans and dogs. And I mentioned humans and dogs because the disease is so similar in both species. So hopefully there can be this reciprocal approach where we learn from breast cancer studies on humans to inform our studies on dogs, and vice versa. We can maybe use the dog as a model organism for humans to be able to answer some tricky questions that are difficult for humans. So, for example, this has been already explored very, very briefly in dogs on a metabolomic level. but they've only been looking at really like characterizing, well, what even is canine mammary tumor with relation to breast cancer, for example. But after characterizing it, then you kind of have to understand and bring in these environmental factors and seeing how these like long-term environmental exposures might influence the incidence and development of mammary cancer or breast cancer. And there's a problem with this. Studying dogs might help this problem, and that is that, well, dogs are terrible liars, whereas humans are not terrible liars. So like this dog is very, very guilty. I mean, so this is something that we can utilize by controlling, for example, what a dog eats. And like Hannes mentioned as well, diet and metabolism go very well, much hand in hand. What we eat influences our metabolism, and it's also been established that metabolism is a huge driving force in the development of disease states, such as mammary cancer. So if we have to understand how diet influences the progression and development of mammary cancer, we really have to know what the diet is. And in studying humans, or previous research groups have decided that we might as well just, we're better off studying mice, because at least then we can know what they're actually eating. And when studying humans, for one, it's very expensive to make a diet intervention study, but it's also even harder to verify and control that the participants are being honest. So then we also have this thing about studying urine samples. And urine samples are non-invasive, they're easy-ish to collect, and they're quite stable. So then we can also be able to explore these things, because urine is the mode, if you think about what urine is, it's like the excess of stuff that's soluble in water, that it gets excreted from the body. So whatever the body doesn't necessarily need, it gets rid of in the urine. So for example, environmental toxins or something of that nature is best suited to be discovered in urine, because that's where it gets excreted out of. So if we're able to, try to see this nature versus nurture component, if we're like thinking about the underlying genes and everything that's built up from that in the organism to have its phenotype, how is that influenced by the environment? So that urine is a good intersection for exploring that. So in any case, we have some relationships between metabolic health and diet, and that's been established in other disease phenotypes, and so we're able to establish that we have the basics already, and there are certain things like inflammatory markers such as some amino acids that are elevated in many disease states, and that was actually Hannes made a very or he covered that very nicely. But in my question, I have two underlying questions that I've been trying to address in this previous study, and that's that mammary cancer in dogs, it's a very heterogeneous disease, by which I mean that even though we could refer to it as mammary cancer, we're actually talking about a whole list of diseases that just so happen to be taking place in the same region of the dog's body, and they all happen to do with undivided cell growth. But the actual quality of these cells, where they started, where they're going, how easy it is for them to, for example, spread in the body and metastasize is highly different. So I wanted to see if there's the different types of mammary tumors that they can be differentiated by the urine metabolites. But then I also wanted to look at the dietary influence and how that might impact my results. So this is a quick overview. I designed the design and sample collection is basically we have urine samples from healthy dogs, and then we have some sample preparation and collection. We have some data analysis, and then we have some future steps that I'll hopefully have time to get into. But in a sense, we had 118 dogs, and we collected a bunch of data from them, both from veterinary offices we received pathology reports for the actual histopathology of the tumor. And the mammary tumor that was in these dogs was hence removed. We collected urine samples from them right before the tumors were removed. And so we were able to not only characterize what type of tumors the dogs had, but we were able to collect the urine right before the tumor was removed, so we were able to see what their urine was like in a fasted state. with the tumor, and then we had a bunch of healthy controls. It was analyzed on mass spectrometry, which has, albeit its flaws, is a pretty cool instrument, and it gives a lot of data. And what it does is that we can get like sort of a snapshot quite quickly about, okay, what's going on here. And this is a confusing picture, maybe intentionally so a little bit of clarification. So we have our disease groups, and they're basically different types of mammary cancers. There's basically from the upper row to the lower row. I really wish I had a pointer, but I don't. So we basically have a control here, and then the idea is that there's like benign tumors here, and then as we go and progress to the right, we can see more and more malignant tumors, or tumors with worse prognosis. So here at the red bar up there, you can see that there's this transition from benign to malignant. And what we can see is that there is a region here that is quite heterogeneous, but there's a little bit more blue here and red here. And there's a little bit more red here and blue here. And what does that mean? Well, that means that here in these disease groups, there's a large group of metabolites that seem to be downregulated, i.e. in lower amounts in cancer patients. But then in some cases, in instances, there's a small group of increasing metabolites. And so if we combine what I just showed here, those classes, and I put them into partial least squares or principal component analysis, we can see that they're actually quite overlapping. And this probably has to do with the fact that they are overlapping. There is a signature of mammary cancer in dogs, and this is, regardless of what kind of benign or malignant, a lot of tumorigenic, by which I mean metabolites involved in tumor processes, are overlapping. So there is a lot of overlap, but there's also, what I found is that there's a lot of metabolites that seem to be doing similar things, and I'll get into that later. But if we think about, like, is there a pattern? Well, I mentioned that heat map, and I said that there's this spectrum from like benign to worse prognosis, more malignant. And if I order them in that way, I say that there is a pattern here. There's a pattern from 1 to 10. Here we have 10 classes. And that essentially, from our histopathological knowledge of mammary cancer, we know that the tumor prognosis worsens here. So what metabolites follow that trend? And we can see that, for example, there's some that seem to be decreasing as the prognosis gets worse. And I highlight two. The one that's most important is probably N-acetylcysteine. And that's because it's a very well-known antioxidant that has, it's quite extensively studied, and it's also a quite popular supplement. So it's quite interesting just to see that that is the most drastic thing that decreases as the type of mammary cancer gets more dangerous. So if we look at the NAC levels, we see that, yeah, it does tend to decrease across the spectrum, and this is quite quite statistically significant. And then we can see that there's this other metabolite called tranexamic acid, which seems to follow this same trend. And we can see that there's actually some subtype differences here in between. For example, in tubular carcinoma, it's actually high or as high as in the control group, but in the case of tubular carcinoma, NAC is actually down. So there's also this type of differentiability. But then, yeah, we can also see on the other side of the spectrum that there's N-acetyltryptophan, and it comes up here twice because, well, I mentioned the fancy instrument that we measured with. Well, we measured with different types of modes, and so the fact that it shows up here twice just means that it was detected in multiple modes, which means it's more reliable. But it just means that N-acetyltryptophan does seem to have this trend of being associated with increased risk of mammary cancer. But I think it's interesting to show that actually, when it comes to N-acetyltryptophan, it doesn't really seem to have this same trend of being linearly dependent on, like, as the mammary tumor gets riskier, the amount increases. No, this is more of a basic tumorigenic process where it's like, regardless of how dangerous the tumor is, you will find elevated levels of this metabolite. So that's also quite relevant. And then we can also think about, like, well, is there maybe a better idea would be to look at just one subtype at a time to be able to get a signal from just one tumor at a time. So I'll just mention a couple, and I'll mention the most common form of mammary cancer in dogs, which is benign mixed tumor. And then after that, I'll talk about ductal carcinoma, which is possibly the most translatable of the mammary tumor subtypes for human research. So if we look at benign mixed tumors, which are, as the name implies, a relatively harmless form of mammary cancer, or well, it's not even a mammary cancer, it's a mammary tumor. We can see that there's some things that very much highly correlate with it. And for example, some things aren't produced in the body, and some things are very much associated with diet. And so, for example, we can see two features here. One of them being caffeine, which I'm sure you're all familiar with, but it was actually news to me just how toxic caffeine is to dogs. I found it very fascinating that caffeine was found in such high amounts in dogs with mammary tumors. It's also interesting to note that it came up as so significant just because there was like a handful of dogs in the set that just had very, very high levels of caffeine compared to the rest, and that's sort of expected because, yeah, you should be giving caffeine to dogs. And then daisine, which is one of the main isoflavones and isolates of soy protein, so it's a very good indicator of soy consumption. So it's very interesting to see just that there's actually, when looking at just the tumor, we can see that there's these implications from diet, but you could argue, okay, well, what about like, you know, using diet as a cofactor, and I'll get into that later. But yeah, we can also see that creatine seems to be quite heavily correlated with it. But yeah, then if we look at like how that plays out and how that looks like on a like more holistic level, what we can see is that, well, most things are upregulated, and there's actually, if you can see within this red square, there's only one row that is actually kind of blue relative to all the other ones, which means that if we have this green bar here, which is all of the dogs with benign mixed tumors, you compare them to the control, then you have this like sort of characteristic of most things being upregulated, and then pyrocatechols 2-sulfate being downregulated. And that's a plant's antioxidant, and I'll talk about antioxidants a lot, so I'm just bringing it up here because of that. But, and then, oh yeah, and yeah, it's also, well, yeah, very interesting to see that vitamin B5 is so heavily upregulated. But yeah, so if you look at something else, we can talk about ductal carcinoma. Ductal carcinoma is sort of a juxtaposition because I just presented one of the least lethal forms of mammary cancer in dogs. And then this is one of the more lethal. It's hard to say what is the most lethal based on histopathology, but this is definitely one with poor prognosis. And you can see that there's actually quite a difference. What I think is quite interesting is that N-acetylcysteine and carnosine are both very much implicated, and we can see here that red means upregulated and blue means downregulated. So in the ductal carcinoma, there's very low levels of carnosine and N-acetylcysteine, and well, hypochloric acid for that matter. And what's interesting with these metabolites is that they're all potent antioxidants. So we can see that, yeah, for example, N-acetylcysteine here is a good indicator of there being ductal carcinoma. Yeah, and then we can see that daisine here actually has the opposite thing, where in benign mixed tumors, it seems to be strongly implicated with being in increased amounts, but here in ductal carcinoma, even though the significance is lower, it seems to be the opposite effect. But yeah, and then we also have the transthymic acid, however, which follow the same trend as benign mixed tumors. Right, so yeah, I think it's interesting just to highlight, if you recall the heat map I just showed you about benign mixed tumors, I had this big red square and all of the values were red in that red square except for this one row of blue. Well, here we have the exact opposite thing going on where pretty much everything relevant between control dogs and ductal carcinomas are downregulated. The only thing that's upregulated is this disaccharide, mellibiose, which is galactose and sucrose. And then we have N-acetyl tryptophan, which is, well, basically tryptophan, but in its excretable form. So you can see that I mentioned the tryptophan seems to be this generic marker that's upregulated regardless of the tumor type. So you can say, okay, well, if there's tryptophan and that's elevated, that's a good indicator of very generic tumor processes. But then if we are able to see that, okay, well, we have upregulated tryptophan, but all of these other biomarkers are heavily downregulated, then we're saying, okay, well, this is probably a carcinoma, a malignant tumor, and not a benign one. So for example, yeah, we had those antioxidants I mentioned earlier. So yeah, this is basically one way of characterizing tumors, but then we were thinking about, I was thinking like, well, okay, it seems that there's a lot of overlap with these different types of tumors, so maybe there's some more general characteristics, like the size of it, or if there's multiple tumors in the body, and maybe that can give some type of a signal. So I basically looked at these same types of patterns of like from less tumors to more tumors, from small tumors to large tumors, and see if there's any metabolites that seem to particularly follow that trend. And we can see that, for example, if you have one tumor or multiple tumors, then we can see that, yeah, N-acetyl tryptophan definitely is highly correlated with the presence of more tumors. And if you look, the only thing that came up as significantly downregulated as the amount of tumors decreases is N-acetylcysteine. And then, yeah, we can see very similar trends. I wanted to also look at, like, well, what if we had a trend of healthy multiple tumors and then just one tumor? This sort of confirms that N-acetyl tryptophan is just always upregulated regardless of the amount of tumors. It seems to be very generic. Same thing with N-acetylcysteine. But then we have this whole group of other antioxidants, and this, well, raises the question that maybe there is some heterogeneity because of malignant processes, for example. Yeah, so okay, I mentioned that this was the number of tumors, but then also, yeah, the size of tumors follows a very similar trend. You have carnitines, I will be talking about them in just a bit, but then also we have this characteristic set, very small, but characteristic set of antioxidants that are downregulated. Right, and yeah, so we've established that CMT subtypes are differentiable, but there's a lot of overlap between subtypes. So if we would want to characterize them with something more than just basically like understanding like how these basic, how these basic processes or like tumorogenic, like generic markers could indicate a tumor, I wanted to sort of explore like, well, what about these antioxidants that seem to be popping up everywhere? And so this is another way of representing what I just showed you in the previous slide on trends, patterns, basically. And we can see that, yeah, okay, there is from no tumor to large tumor, there's N-acetylcysteine and transthymic acid that seem to follow this trend of decreasing amounts. And that's rather fascinating. It seems that there's almost like a safeguard mechanism that's lacking for these dogs that are suffering from large tumors, or the large tumors themselves seem to be requiring a huge boost in antioxidant status. So yeah, and then this is looking at the malignancy of tumors. It doesn't seem to be as correlated with malignancy. So this is also referring to the fact that it's a generic marker and not something specific to a subtype. So yeah, I have promised to talk a little bit about diet, so I'll be using my final 5 minutes to talk about diet. And there are some things that I've established in previous work, in previous metabolomic studies. For example, methionine, and methionine is an interesting one because it's this very potent growth signaler, and high levels of methionine have been associated with low lifespan or short lifespans, for example. Restricting methionine is one of the well-known ways of extending lifespan, so it's just very interesting to see just how upregulated it is in kibble groups, and even in the case case dogs that were eating raw, it was less, which is interesting because cancer is thought to be an inflammatory disease. Methionine is a marker for many cancers. Yeah, so we were able to combine the data from mammary tumor and from diet. And here I'm talking about, I'll describe the groups quite briefly. Yeah, so we had basically this raw group that was eating a majority raw diet, and that had to do with the setup of our research questions in the first time. We weren't looking for raw fed dogs in particular. They were just the dogs that happened to appear in our study. So yeah, we have we're comparing dogs that ate 100% kibble. Finding dogs who ate 100% kibble was very easy, so we definitely use them as control for all of these. And finding especially dogs with mammary cancer that had eaten raw food was very difficult. So unfortunately, we had to settle with this majority raw group instead. But yeah, it was very interesting to see that if we compare the healthy dogs, so here's control raw-fed dogs, control kibble. So these are only healthy dogs. You can see that there's this region here of red and a region here of red. And then this is the raw-fed dogs. And you can see that there's this list here, and that's all upregulated in the raw-fed dogs. What's interesting is that these are all the carnitines, and carnitines basically have to do with providing energy to the mitochondria for from fatty acid. So that's the only thing that's upregulated in healthy dogs. But then if you compare the dogs that have mammary tumors, so here we're looking at the raw-fed dogs, the kibble-fed dogs, then yeah, we can see that there still are some carnitines that seem to be involved, and that there's some things that were previously associated with, for example, benign mixed tumors that is significantly lower in the raw-fed group, such as hypogalic acid and diazine, which is the soy isolate. So it's not surprising to see that there's far lower amounts of soy products in dogs that have been fed in a diet higher in raw food. Yeah. And then we can see also that methionine levels in the dogs that are fed raw food are significantly lower in also the dogs with mammary cancer. So yeah, and if we then look at just the dogs that are being fed different diets, but they both have cancer, you can see here is the things, the metabolic pathways that are especially relevant to that, and by far the most is sugar metabolism. So the processing of carbohydrates is what differentiates these dogs by far the most. But it's also interesting to see that, for example, the oxidation of branched-chain fatty acids is something that's significant, and you would almost expect it to be in the raw-fed dogs, but it seems that it's in the kibble-fed dogs where that's increased. Yeah, but then if you're looking at more of the difference between healthy dogs and mammary dogs, and here just to show that there was actually the most difference between healthy raw-fed dogs and dogs that were eating kibble and at mammary tumors, and that's unsurprising because there you have like both the influence of diet and mammary tumors. We can see that tryptophan metabolism is the most relevant to this, but then also fatty acid biosynthesis and fatty acid elongation in mitochondria. So yeah, you can see that there's some different things. And then so I've talked a lot about antioxidants, and antioxidants are typical, and polyphenols especially, are something that were downregulated in dogs with these severe forms of, or more dangerous forms of mammary cancer. So it's typically thought that, antioxidants is something that you get from plant-based foods, but I would argue that it's actually the processing itself that will determine the antioxidant status, where, for example, if you have something that is heat-treated for a long period of time, the antioxidant status is going to decrease. And this myth, for example, that meat does not contain antioxidants is only true in the case of cooked meat. Like if you have very highly processed meat, then yes, you would expect lower levels of antioxidants, but it seems to be that there's this actual big processing component that might have more to do than the actual meat or plant content. So that's one thing, that's one take-home message, but then it also is interesting to show that benign tumors seem to have this characteristic upregulation of most things, and there's like hardly anything missing, whereas if you have malignant cancers, most metabolites are down-regulated, and there's only a couple of these characteristic metabolites that are quite generic, like tryptophan that I mentioned, that are upregulated. So future research is made basically to trace this path from the start to the finish, and I unfortunately do not have time to go into that. But I want to now trace that, like I've been looking at urine, so I'm starting with the tumor, and then looking through the actual tumor itself, blood, and then urine. So with that, I would like to thank all of the people who have supported me throughout this journey. It's been great. And Alessandra, my supervisor from Brazil, who is a histopathologist, was very kind and characterized all of the tumors for me. And yes, thank you all for listening.